Thank you so much. So yes, it's actually a cute little Latin word for a type of frog that lives in Africa, but it doesn't really matter for the talk. All right, so I understand that biochemistry, molecular biology isn't necessarily everyone's strong suit, so I tried to punctuate my talk with lots of random pictures of mostly my children spending time in Bavaria. So you can look forward to that as this week goes flying by. All right, so during fall 2014, I was on sabbatical. Thank you all for letting me go. I was here in Munich. So this is looking out from St. Peter's Cathedral, St. Peter's Church in Munich. This is the new town hall. You can see the Olympic Stadium, Olympic Tower in the background. Here's the royal residence from back when Bavaria was an independent kingdom. If you have really good eyes, this little white blurb here is the football stadium where the two teams play. This guy looking up toward where my apartment was. I was working with Dr. Felix Müller-Pence at the Ludwig Maximilian University in Munich. This right here is actually the main campus of that, but we were on the medical center campus. And I'll show you a map of where that was later on. In case you don't know, here's Western Europe, here's Germany, this big huge looking thing. Munich is way down south in Germany. And it's in the state of Bavaria, which for a long time was an independent country. And so the people in Bavaria, and in Munich in particular, tend to have their own way of doing things, which makes it kind of an interesting cultural experience being there. There's a little wonderful zoom in of Munich. Where the N is in the word Munich was where that picture was taken from, I just showed you. I worked really close by the medical center, and I lived up by the soccer stadium. If you turn right from that previous photo, you're looking out to the west, I'm sorry, to the east now, over beautiful Munich. This is the old city of Munich. I show this shot because right here is a very good museum called the Deutsches Museum, which is actually worth a trip to Europe simply to go there. John's nodding his head in agreement. If you've never been there, for the love of God, you need to go see this museum. It's a great museum for physicists because everything is there and they want you to touch and play with everything. So it's fun. Okay, so turn the other way. So look now to the west, main train station. Here's where our lab was, and for those of you who like to drink, this is where the Oktoberfest grounds are. So we weren't too far away. Okay, so a very simple way of thinking about how life is organized is you have mainly two big classes of molecules you deal with. One is DNA. As you know, this is the molecule that carries genetic information for your cells, for you. And specifically what it carries is the information that is required to make proteins. Proteins are molecules that do things. So they're involved in stimulus response, they're involved in replication, they're involved in structural work, they're involved in different kinds of catalysis and metabolism, these sorts of things. And there's an interplay back and forth between the DNA and the proteins. The DNA contains the information to make the proteins, and the proteins themselves can manipulate the DNA, up with its storage, with its replication, etc. So there's a back and forth that goes on. DNA is a polymer of nucleotides. And you've probably all seen these in science fiction movies before, the A, the T, the G, and the C. So they are nitrogen-containing little rings molecules around. And if you take one of these little rings, like cytosine, attach it to a sugar into a phosphate group, string them all together, you make a polymer of DNA. And it's the sequence of these A's and T's and G's and C's that tell the cell how to make proteins. So within that code is that information. Proteins, on the other hand, are a polymer made of amino acids. And the different kinds of amino acids, there are some that are acidic, some that are polar, some that are hydrophobic, some that are hydrophilic. And they have different little properties here. They're all relatively small, little carbon, nitrogen-containing molecules. And the proteins are made by stringing together a series of amino acids, which then will fold and compact upon itself to form the correct structure it needs for its function. Jeremy Schmidt from K-State was here a few weeks ago. He gave a talk in our program, and part of it was about this protein folding situation. He does simulations working on that. I'm not going to talk about that at all, really, except to let you know that when proteins are made, they're made as linear polymers, but they must collapse down to some final folded structure for it to function. One of the goals of molecular biology is to have structures like this because if you can see what the molecule looks like, it gives you insight into how it actually works. Unfortunately, a lot of molecules are very, very hard to get into a form where you can visualize their structure through crystallography, through some kind of electron diffraction, etc. Okay. DNA. So we're familiar with DNA being this double helix molecule that Watson and Crick found out. If you were to stretch out the DNA that is in your cells, it'd be about 1 meter long. Your cells are somewhere in the order of a micrometer in length. So there's a problem with fitting the DNA into the cell. So what our cells do is they package the DNA into a very compact form so that it can be shoved inside the cell or inside the nucleus of the cell. So it gets wrapped around other different kinds of proteins, and those little globules get stuck together, and they form different structures. They all have cute little names, and eventually it forms this thing that you've seen before, probably called a chromosome, which has this little X shape to it. And the chromosomes are what get finally stuck inside of the cell. This whole process of this kind of packaging, making chromosomes in the DNA, is called chromatin. There are different kinds of chromatin. We'll be discussing one kind here, but overall, that's the term that's given to this hierarchical folding structure. There's another picture about this, which is supposed to give you some kind of a scale. I take the DNA, then I sort of wrap it around these other protein molecules called histones to form a beads on the string structure. That gets further packed and condensed and down to finally form a chromosome. I'm interested in this level of compaction, the first level of compaction of the chromatin, which is called a nucleosome. So I take the DNA and I wrap it around an octamer of proteins, which are called histones. And I wrap about 150 base pairs of DNA around each of these octamers to form these nucleosome structures. So this is the first level of compaction of the DNA, and it's kind of what I'm interested in figuring out. How does this work for wrapping and unwrapping the DNA around these histones? So these histones are structural proteins, so their job is simply to serve as a scaffold for the DNA. They don't do anything, yes?
That's just one strand of DNA going around the histones?
Yeah, so we have some, roughly meter long piece of DNA. Parts that get wrapped around here, 148 base pairs around each of these things that goes on.
That's just one.
One strand of DNA, one big long strand of DNA. That's a double helix of DNA,
Right.
but it's one long double helix DNA. And get shoved out there.
There's no animals that have two or more.
Animals that have what?
No instances of two distinct DNA strands.
Wrapped around the same histones?
Just double this. Well, you've got, I guess I meant, each chromosome has a strand of DNA in it.
Right.
We have 21 chromosomes.
Right.
There's 21 copies like this. All right. You guys here think there's one long DNA, but there's a bunch of long DNA.
Yes, but there's only one that's being wrapped around these nucleosomes in one particular segment of nucleosomes. It's not multiple than that. So packaging the DNA into the chromatin is a very good thing, mainly because it allows you to sequester all the DNA into the nucleus of your cell. The nucleus of your cell is even smaller than the cell itself. So I really got to wrap the DNA up a whole bunch to shove it in there. By wrapping it up and shoving it in the nucleus, I can protect the DNA better. I can keep it free from a lot of oxidizing agents or other kind of things that can mess up the DNA. The problem with packaging DNA into chromatin is that it makes it more difficult for you to access, for the cells to access the individual genetic information contained within the DNA. So that sequence of A's and T's and G's and C's is harder to read when the DNA is all wrapped up and packaged. So cells have evolved several different mechanisms that allow them to go back and forth between having the DNA all packaged up and secured and having it unpackaged so that the information it contains can be read. One mechanism that does this depends upon the function of a certain kind of protein in your cells called a chromatin remodeling protein. So again, chromatin is the package of the DNA, so the name chromatin remodeler means it's a protein that reorganizes or remodels the chromatin. These remodelers are frequently noted as molecular machines in the literature because they require an energy input to do their work of remodeling or reorganizing the chromatin. And the energy they get is chemical potential energy from the hydrolysis of ATP. But it's still not exactly clear how these things do their task, and I even quibble about whether or not they're actually using the free energy in that way, so it's a little bit of a sort of a different story. Questions about this so far? They go flying through. So, my work with Felix was focused on these chromatin remodeling proteins interact with nucleosomes, the first level of chromatin structure. This is a crystal structure that was made of a single nucleosome. The multicolored bits in here are these histone proteins, and this is the DNA wrapped all the way around it. So the question that we have is, how do these chromatin remodelers bind to the structure? Like, what are the individual atomic contacts that are made? And then how do they then get the DNA to be moved relative to these histones? How do they cause that translation to occur? Or how do they cause histones to be ejected? And then what I'm kind of interested in is how efficient are these remodeling proteins at doing this task? Like, how much ATP must they consume to get this done? How good are they at the energy conversion process? So that's what Felix works on, that's what I worked on, so that was our plan to work on that a little bit. Okay. So first of all, I'll take a little break, because that was a lot of chemistry. So in 1810, okay, the Crown Prince of Bavaria married Princess Teresa of Saxe, that's a wonderful word, isn't it? Hildburghausen, all right? Now, they had this big, huge wedding ceremony. It was a big, huge deal, because he was kind of a famous bachelor at the time. And so they organized these huge festivities right outside the city gates. And what they had as the main part of this was a bunch of horse racing. And everyone was invited to attend. It was a big, huge, fun thing. And so it was so popular that they decided to have the horse races again the next year. And then the year after that, and then so that became Oktoberfest. Oktoberfest is basically a state fair, is what you think about it. So there's a lot of livestock showing, a lot of horse racing, this kind of fun stuff. But recently, it's been known for basically one thing, and that is alcohol consumption. So here is a look at Oktoberfest, looking down from the Ferris wheel at the various beer tents. Now the word tent, I think, is kind of loosely being applied here, because these places hold on order of many thousands of people. So it's a little bit on the insane side. 5,000 to 10,000 people inside a tent. So the different breweries, there are 6 breweries in Munich, and each brewery is allowed to have a tent. And then some of the tents kind of show up here and there for other reasons. You have a bunch of carnival rides you can go on. There's a bunch of other little food stalls, stuff like that. But it's mainly a bunch of people milling around, trying to get in here to drink a lot of beer. If you get a little further down, you can kind of get a better view of the crowd. This is looking down sort of the main street there. A bunch of people. People who are exceptionally drunk in the picture tend to be Australians. And a few British people. It's kind of funny. It does. It was a cultural experience. Now, what's interesting about this is that in the weeks leading up to Oktoberfest, you begin to see people wearing the Tracht, the costumes, more and more. Now, you see people in Bavaria. We saw them wearing the lederhosen the whole time we were there. But the locals start to get it out more and more and more in the weeks that get up to Oktoberfest. And people who show up to start drinking also go buy some and stuff like that. So it kind of becomes a fun thing to be wearing all this clothes all the time. Since we were trying to get ourselves more blended in with German society there, of course we had to partake in that as well. So here are my lovely children wearing their dindles that we bought them to go over Oktoberfest in. And they got to go downtown and hang out Oktoberfest and see all the drinking and frivolity. All right, so Oktoberfest is fun. Now, back to science. So we worked with a particular chromatin remodeler called ISWI. Now, one thing that's so much fun about biology is that they have this penchant for making up acronyms for different words, or acronyms within acronyms, and has this weird lore behind it that usually makes absolutely no sense. So this stands for an imitation switch protein. Now, first of all, you gotta know what a switch protein is, know why an imitation switch protein makes any sense. But don't worry about it. We just call it ISWI. ISWI by itself is a protein, and it's capable of doing this little repositioning, rearrangement of nucleosomes all by itself. There are homologs, which are genetically or evolutionarily related proteins for ISWI, found all over the place. Fruit flies, plants, worms, frogs, you name it, humans. We all have some thing that looks like and works like ISWI. And these different homologs are really, really evolutionally well conserved. So the ISWI you see in a frog is similar to the ISWI you see in a human, etc. Now, of course, the biologists give them all different names, just to make it confusing, but they're all basically the same protein. ISWI also serves as a functional component of many other larger complexes of proteins that also remodel chromatin. So in this picture, which you cannot possibly make out, all of the red circles are ISWI proteins. So here we have yeast, worms, some other kind of bug thing, plants, fruit flies, humans, and frogs. Everywhere you see a red circle, it's ISWI. It's in many different kinds of complexes, including ones that, well, they don't know what the other guy is, because it's a question mark. But it's in many different kinds of complexes, and a lot of these complexes do different things with chromatin, but they're all involved in some aspect of chromatin remodeling. And some of them are targeted differently, some are more important when cells are replicating, or some more important for damage, et cetera, et cetera. But they all share this sort of fundamental little component. So we were kind of curious to learn more about how that little component works, because if we can then figure that out, we can have a better idea of how some other things might work. Okay? So the idea is, again, we have this nucleosome structure, the DNA wrapped around these histone proteins. And we want to know, how is it that ISWI somehow binds to this structure, interacts with this structure, and is able to move this DNA relative to the histones? It can move to a translational shift of those. How is that possible? So to begin, we can talk about, why does anyone care about this? So because your DNA gets packaged up into this chromatin, it gets sequestered away inside the nucleus, and it's hard to access the information that it contains. So if you need to copy your DNA, for example, you need to undo all the chromatin so the DNA can be copied. If there's damage in your DNA, you need to undo the chromatin around the damage to fix the damage. If you eat a lot of chocolate, you need to make some more insulin, right? You need to somehow find the insulin gene, unravel that bit of it, and express that protein for yourself. So the cells need ways of moving histones around and organizing chromatin in general. A lot of the models you find are kind of what I call this sort of cartoony stage. And the reason why is that it's very difficult to get structural information about a lot of these proteins, because they're kind of floppy, or they're inflexible, or they're hard to grow in large quantities, etc. The nucleosome was a feat in that by itself. Some people argue, okay, well, here's my DNA, here's some nucleosome. Maybe what happens is my protein, my remodeling protein, binds to part of the nucleosome and has some other arm that kind of sticks out, and that walks along the DNA. And as it walks along the DNA, it can make some kind of a loop or a bulge, and then that gets magically propagated around the whole octamer, and that's how these things work. All people have argued that no, what happens is your protein here in yellow binds to this nucleosome. When it does that, it competes with the histones for binding the DNA. So it basically loosens up this whole structure of DNA and histones. And when that happens, it's very easy then for the DNA to kind of slide around, because it's not so tightly bound to the histones anymore. Recent results from our lab, before I left my sabbatical, is that we discovered that a single molecule of ISWI by itself was sufficient to do all of the work of nucleosome repositioning that we could detect. And that was interesting because there's a lot of arguments in the literature based upon faulty data analysis. But there's a lot of arguments in the literature that argue that it's 2 molecules that are required. We argue it's only 1. And this is interesting because we know that a single nucleosome can accommodate up to 2 ISWIs bound at a given time. So you can bind 2 of these things, but only one of them seems to do any work. We also looked to see how efficient this molecule was, meaning how much ATP is required for it to do its job. And what we found was a lot of ATP was required for this molecule ISWI to actually move anything around with regards to the nucleosome. And we conclude that's probably because there's a lot of sort of futile starting events trying to get this process started and end up failing. And there's more we can talk about. I discuss about that later on if necessary. So we have this sort of interesting data, and so the thought was we'd like to pursue this a little further with a sabbatical joining with Felix in Munich to say, can we figure out anything about the details of the interactions between ISWI and the nucleosome? Specifically, can we get any kind of structural information so we can figure out how are these two different ISWI molecules bound to the nucleosome? What does that tell us about why only one of them seems to be doing any work? Or what does that mean for how efficient they all are? Can we infer any kind of even mechanical mechanism for nucleosome repositioning based upon how these things are bound, etc.? So, there are no structures that are currently known for ISWI, no crystal structures, any kind of high-resolution structure that exists. So people have tried to determine different kinds of models for ISWI functions based upon simple mutagenesis from experiments. I will literally remove big chunks of the protein ISWI and see what happens. Oh, that killed the cells. That must be important. I'll put it back in. I'll take this part out. Put this part back in. So when people do that over and over again, they get these more cartoons about how, well, I think there's something that's here, that's blue, and something that's green over here, and this like mechanical hinge connects them somehow. And this is how it works. And so these sort of models get on, but there's really no good data to support them one way or the other besides mechanistic data. So what Felix and I wanted to figure out was, is there a way we can try to get at what the structure might be, rather than trying to go through crystallography, which is not working, or these kinds of experiments which don't seem to give us that much information. And the thought was to build on his experience doing a technique called chemical cross-linking. So remember that proteins are polymer nucleic of amino acids. So there are different kinds of chemicals called cross-linkers, which form cross-linking bonds, disulfide kind of bonds, between different amino acids. So if you have two proteins that are kind of close to each other, if you can get them to be cross-linked, you can sort of lock their configurations together. And that's a little more easy then to look at what their structure is, what they're actually doing. So what we do in Felix's lab was first we try to cross-link them using a specific kind of chemical that will attach different amino acids in these two different complexes. And then we'll chew the whole thing up and feed it to a mass spec to see what parts come out. It's a little bit like quantum physics. So since we tend to know the amino acid sequence of a protein, even if we don't know the structure, by looking at what parts are linked together after the cross-linking, we have some idea as to how the proteins are put together. So imagine I have two proteins, and here I've listed the proteins in terms of their amino acid sequence using the simple one-letter code that we have for amino acids. Anyways, if I cross-link these two together and chew them up, maybe I'd see a fragment that looks like this. I'd see LCA cross-linked to RCD, where the two cysteines are cross-linked together. If you knew this was something that existed in your complex, and you knew that these two proteins were fed into the system, you would know that you must have had something look like this that actually got cross-linked. When proteins come together, this is how they were kind of oriented relative to each other.
So you know that C is attached to C, but do you also know that L is closest to I?
Right, but you would know, you could detect the actual cross-linking in your mass spec. You could see that, you could see this fragment together, so you would know, yeah, you know these are all together like this in somewhere.
So it's not like RCD could be sort of switched.
Yeah, it's possible that could be switched around.
It could be D, could be.
You could have this be ACL, and this could be, DCR. That gets, we'll talk about that in a little bit. So all you can figure out is roughly, this is where there's some connection point in my system. Now, here's a slide to show how this works out. Here's some big horrible thing. I'm trying to show you the different colored components of some big protein complex. They added some chemical cross-linker, which is some tiny little chemical like this. It forms cross-links all over the place. Proteolysis means you chew up the protein, make a bunch of little tiny segments of it like this. And you can see sometimes I've got red and blue linked together. Here's red and red linked together, etc. You feed it to a mass spec, and it spits out a measure of the charge to mass ratio of the different components you might have in your system. And from that, you can do some kind of really cute computer work to figure out, well, what must have been the contact points in all of my different proteins to have arrived at that pattern of cross-linking. And then from there, you can develop different structural models to explain what might be going on. You can say, well, these two are consistent with this mass spec data, but some of the other ones aren't. This really part of the problem here is that mass spec, and this is just a horrible little quick Wikipedia stolen figure of a mass spec machine. Okay. The idea is that you have some kind of method of ionizing your samples after you digest them, and then you separate them out based upon their charge to mass ratio. And so you read out a measure of abundance of different charge to mass distributions you have for the samples you fed into the mass spec. The problem with this is that this data is not very specific to a specific kind of configuration of molecules in your system. You can have many different combinations of amino acids in different ionization states that have the exact same charge to mass ratio. So your data analysis gets really, really messy for really large complexes of protein, because you have so many possible combinations that can give you the data that you're looking at. It's hard to know which one is correct. Plus, most of the cross-linkers aren't very specific about what they actually cross-link. So this makes the data analysis even more complicated because you don't really know what you're looking at all the time. Because you could be cross-linking a bunch of stuff together, not specifically targeting different things. So for example, let's imagine you have a simple complex where protein A binds protein B in this configuration. If you're super lucky, you have your cross-linker in, and you get some simple cross-link between A and B, maybe some internal cross-links, but not so bad. But because of science, and the way science works, you tend to get just goop. Because the cross-linker cross-links everything together, and you get this disaster that comes out. And when you chew this thing up, you get all kinds of possible connections, and who knows what's what. Yes?
Yeah, but because of this, can't you just run a gel and just clear them out that way, by mass?
So you could take these things and run a nature gel. The problem is that makes it more difficult to feed into the mass spec later on. So it's simpler just to purify them sort of directly from this. But yeah, you could do that. But even then, again, the gel is going to only, you could rule out huge amounts of goo. But if you've got small little bits of goo, they can migrate the same way as things that you're interested in. So how would you know what's what? Plus, the resolution of the gel might not be the best. But yes, and that's foreshadowing one of my little insights later on I had for them. So to make life simpler, because that's our goal in life, should we make life simpler? We wanted to use specific chemicals that would cross-link only specific pairs of amino acids. So there are a lot of cross-linkers which cross-link a bunch of different amino acids altogether. We want to specifically cross-link just a few of them. To make this work, we had to make sure that our protein of interest, ISWI, contained only a subset of possible targets for these cross-linkers. So I spent a lot of time making mutants of ISWI that contain specific cross-linking targets. So I would change the sequence, the composition of the ISWI proteins that had only one of the amino acids I was going to try to cross-link. And we could put that amino acid at different locations in the protein and then see, do any of these sort of things line up with the complex? And then we could use specific chemicals to cross-link these mutants to the nucleosome. So the idea is something like this. Again, this is the complex that you want, some AB together. Let's imagine you can put your specifically targeted cross-linker here. Then when you form the complex, lo and behold, if it cross-links that way, you'll see the cross-link in your complex. You'll see A is cross-linked to B. And you know, okay, that must have meant that somewhere around here, there was a contact point between protein A and protein B. I put my cross-linking target up here, I mix them together, and I do the mass spec. I'm not going to see A and B ever cross-link together. So I'll know that this interface here is not an interface where A touches B. And similar if I put that somewhere else. So that's the idea. So we make lots and lots and lots of mutants of ISWI, try many different kinds of chemicals for cross-linking. Yes?
The crosslink has to be strong enough so that it survives when the proteins are
Yeah, it's a covalent bond, so it survives.
Then how do you know that the crosslink doesn't substantially initiate new configuration? It's the strongest thing around that can just bend everything into a whole new shape.
It's entirely possible. So what you try to do is another part of this is you try to limit the exposure time of your samples to the crosslinker, so there's not enough time for them to really make everything gooify. But there's a fine line between how much time is enough for it to work, how much is too much that your whole sample is black. So you have to map out those conditions for the experiment. And of course, there's always noise. So you can get data that has, protein that has no cross-linking in it, or it cross-links with itself, or A cross-links with A. So that you have all this other data you got to sort of sift through, looking for the different kind of complex information you're curious about. So there's a lot of trial error, and it's mostly error, which is generally true for most of biology work. You spend a lot of time trying to troubleshoot stuff that tends to never work. And eventually, if you're lucky, you stumble across something that actually is functioning. Okay. So while I was doing that in the lab, my children were enjoying the finer parts of Bavarian life. So Bavaria is known for incredibly large pretzels. This is to scale with my daughter's head and body. And wonderful quantities of alcohol. If you are a child, you can drink with your parents in public. That's okay. You just can't buy alcohol on your own. So this is perfectly legal. And it's no crime you can do.
Herself?
What?
Did she drink that all?
But this one over here is pretty good. I'm proud of her. She can put back at least a half liter now. And they went to Neuschwanstein. It's absolutely gorgeous down there. So much fun. Okay. So while they're off drinking, eating pretzels, and seeing the sights, I was in a cold room in the lab where, ironically, they also store alcohol. This is another fun story. They have no problem whatsoever storing bottles of beer in the biological cold room at medical school. And when I asked about this, meaning, isn't that a violation of every possible health and safety code, I was told, if you want a beer, there's a container in the department office. Just put €1 in there and get yourself a beer. And I said, no, I don't want one. I think they should be next to, say, the yeast samples. No problem. Different lifestyle. Okay, so what do I have to do? So I'd have to get the DNA for the protein. Remember, we can make mutants of this protein. So I have to take the DNA that codes for ISWI, change it a little bit so it makes the mutant form ISWI that we want. We've got to stick that DNA into a cell, make that cell grow the protein for us. Then we've got to harvest the cells, get the protein out, purify the protein, make sure the protein is still functional, because we've changed it around a little bit. Does it still work? Then do all this screwing around that John alluded to, trying to figure out what is the best kind of cross-linking compound for this, what kind of concentration should we use, how much time for the exposure, all this stuff. And do this over and over and over again with many different kinds of cross-linking compounds, many different kinds of mutants, and many different kinds of solution conditions. Change the pH slightly, change temperature slightly. And then if anything envelope was working at all, we would send over to the mass spec lab to do their analysis. Their data would come back, and we'd just sit through all of that and see, is any of this at all meaningful? Did this a long time. After about 10 weeks, I was convinced this was never going to work. So I was kind of new to this process, but I was pretty sure this was sort of doomed from the beginning. So I found out many sort of believable cross-links between ISWI nucleosomes, but nothing was really holding very rigorously when we did the actual mass spec analysis. And a lot of the ISWI mutants were just not very happy. So they would not be, the cells would not grow well with them. They were hard to purify. So as I went through this whole process, I was trying to think about what can I do to make this better, how this work out. At the same time, I made several interesting cultural observations about my coworkers. So the research lab I was at was in the university. It was mostly Germans, but a lot of the grad students were from other places in Europe and other places around the world. One thing that was kind of fun was that whenever we would talk about science, it was always in English. And I thought this was just because at the Institute, not everyone knew German because they were from Japan, or they were from Poland or whatever. But more than once, I'd be in a conversation that involved only German people, native German people. And whenever the topic was about science, it was always in English. But the second we talked about anything personal, it switched to German. And it was really a lot of fun because we go to dinner and we'd switch back and forth really rapidly. So we talk about that day's work in the lab, it's all in English, and then all of a sudden it'd be questions about your family, what you're doing today, and that switches over to German. And then once it gets back to science, it's back to English again. And it oscillates back and forth. I found that just amazing. And I got pretty good at that eventually, and it really helped out if you just drink a lot, I found it. That makes it very easy for your brain to switch back and forth between languages, because you don't drink too much, but just a little bit, and it's really easy. And how much, you say, is it how much? There are rules about this. When you are at lunch, you drink no more than ½ liter of beer. And I learned this. If you don't drink at least ½ liter, they think maybe you're not feeling well, okay? If you go beyond that, they're suspicious that you're up to something. And the rule dinner is only 1 liter. So your goal in life is to basically get a lunch and dinner date on the same day, then you get 1½ liters of beer on the company's dime. So that was kind of fun. Nothing is really interesting about the German people. Julia speaks German. Yay! So, there are many different dialects of German. And I was taught this when I was living in Germany in college. And what I found interesting is that for a little while I went and I worked in the Rhineland. I was living outside of Cologne. And there's a lot of French that gets mixed into the German a little bit. And so you pick up like a slight little, not a French accent, but a sort of Rhineland-y kind of accent, which sounds like it has a little extra French in it. Bavarians, I think this says it all. I think they enjoy being different from other people. And I think it's, again, because they were an independent country for a long time, they kind of just have that mentality. So in English, we say good morning or good day to meet somebody. If you're speaking standard German, you'd say guten Morgen, guten Tag, right? In Bavaria, they say grüß Gott, which literally means may God's greetings be upon you. And I think this stems from the fact that for a long time, right, Bavaria was this bastion of Catholicism against the Protestant Reformation. So they take a great pride in this sort of religious definition of their citizenship. So that was kind of interesting. Okay, it's also, Bavarian is not like Standard German at all in many other reasons. And one thing is that they have a lot of Latin stuff that kind of gets smooshed in there a little bit. So in English you'd say, I like you, I like Munich. In German, ich mag dich, ich mag München, not too far off. In Bavarian you say, ich mag die, ich mag Minga. Now how exactly these things are related I don't really know, but it's kind of, something different there. But what's more fun is if you look at the motto for the Bayern Munich soccer team. So their slogan in English translates as we are we, which is supposed to mean like we are the team, we are all together, that kind of a thing. So in German you would say wir sind wir. But in Bavarian they say mia san mia, which is the sort of Latin-esque weird bastardization of stuff. So more than once we'd be out around town and we try to teach our kids standard German, and we'd run across people who'd be speaking Bavarian, and needless to say this did not go very far. The favorite was when my wife told me the story where she was at an Italian restaurant and she was an American speaking German to an Italian speaking Bavarian. Needless to say, this did not get very far at all. Luckily I was at work. Okay, so after drinking a lot and thinking about languages, that's always fun, and getting myself distracted, I came back to this problem thinking, I at least know what part of the problem is. And it's sort of what John was hinting at. The idea is, this is what you really have in solution. Let's imagine we have a complex that can be 2 A's and a B, or it can be a single A and a B. So what we were doing is we were mixing together all of the A's and the B's together. We let it sit in the tube for a little while till it comes to equilibrium. And then we go through the process of adding in the cross-linker and doing all that kind of stuff. But of course, when the sample is at equilibrium, I have all of these species present in different combinations. And the amount of each one of them depends upon stuff like solution conditions, initial concentrations, etc. But all I really care about maybe is this one, or this one. But if I add cross-linker to all of this stuff, what I get is an unholy mess. Because I get every possible combination of everything with everybody else. And so I get basically this huge amount of noise for very, very, very little signal. Whereas what I really, really want is maybe just this. This becomes a tiny fraction of my total population of goo, and so it's really, really hard to see that. So my proposal to Felix was, why don't we try to see, as Ray was suggesting, can we somehow maybe isolate one of these guys ahead of time, and just target that by itself with cross-linker, so we can get a population of only that which we're interested in. And so you can do this using a process called electrophoresis. So in this experiment, what you do is you load your proteins into a polymer matrix, which we call a gel, and you use an electric potential difference to push the molecules through the gel. As they move through there, things like their shape, their mass, their charge, etc., affect how they move through the gel. So you can use this electrophoresis technique to isolate out individual species of the different things you're looking for. And we've done this before in our lab. We have an experiment where we're looking at ISWI, this protein binding to nucleosomes. So here is this polymer gel that we've imaged. We used a dye that specifically stains DNA to look at the different complexes. So here we have free DNA, here we have the nucleosome by itself, and here we have nucleosomes that are bound with one or two ISWI molecules. So my thought was, why don't we do something like this again, and then we could maybe cut out one of these little bands with a scalpel, take that little bit of the gel, stick it in the crosslinker, and hopefully get just the species we want to be cross-linked and see what happens. And my thought was, they didn't really buy this at first, but my thought was, like, if you have a little bit of luck, this will work. When you're in Munich, if you need luck, you go to the residence. Outside the residence, which is where the king used to live, there are a series of statues of little lions there, and you rub the faces of the lions as you go by for good luck. So we all went there. I made them do this. So we rub our little lion heads here, and then we go on to our experiments. And I was able to show that it works, but even after all of that, we still had to go through the process of trying to figure out what's the right cross-linker, what's the right conditions, all this kind of stuff. So I was not able to get any of that done before I left. So I turned the project over to a grad student and said, here you go, work on this. And unfortunately, she is still working on it. I think it's become, I think Felix has decided this is not going to go very far, so it's become a low priority for him. But she's still working on the same mutants that I made, looking at different cross-linkers and going on further on from there. So with a lot of luck, it might still end up someday in a publication somewhere. So here's a very pretty picture in the Alps there. I like this picture because it's hard to tell what's up and what's down because of the reflections. So I asked my girls to stand in front of it so you can tell which way is which. Okay, so besides drinking and learning German and working in the lab, what else did I do? So there are many times, so in biology land, there's many types of experiments you do where you set stuff up and you have many hours to yourself before you gotta go look at something else. Or you have a whole day with these dialects and whatever. So when those things would happen, I would try to do other interesting stuff. And I went and attended several different lectures in physics and in biology. And it's hilarious because based upon my data set now of two, I have a conclusion. And one is, it's very easy to find the physics book on any campus. You simply go to campus and find the most rotted, old, decrepit building that no one cares about. And that's physics. You want to find the med school, you go find whatever is the nicest, neatest, marble encrusted building, and that's what they are. So it was kind of fun. I talked a lot about our course transformation work. There was some age bias, and whether or not they thought this was a good idea or not, and some discipline bias. The physicists thought this was all hokey and not worth the effort. They'd much rather have their 10,000 seat lecture hall and what they're doing. Whereas the biochemists in the med school are much more interested in this kind of stuff. Part of that, I think, is because, biology and biochemistry lends itself better to these sort of compartmentalized little sort of group work activity kind of things. There's not so much background framework that you build up for that. That was kind of fun. I had a lot of talks with them about how we fund life sciences research in the US versus Germany and what the priorities are in the different countries. That was very interesting. Basically, the Germans have an approach that it's more important to fund, they try to optimize total people being funded, even if people are getting less money, rather than a few people get a lot of money, which is the US model. So that's kind of interesting. Their method tends to favor a lot more collaboration. So everyone in the department I was in was basically working on similar projects. so I can pull resources together. That's kind of interesting about. I think that's something that, if it could be important in the United States, it'd be good. There are many advantages of having collaborative research, especially when you can share equipment. Allows you to have better maximization of productivity. You can form new projects much more easily. The problem with this is that you need to have people in charge of maintaining the equipment. You can't have it be some random thing. The Germans, not surprisingly, were very well organized at that. So worked out fine. And I also finished writing my textbook, which is this thing, which I wrote. So I've been using this thing now in 211 for the past year. So far, pretty good responses. Part 2 on E&M will come out this summer. I'll start using it this fall. Anyone wants to play is more than welcome to. And lastly, I was able to flesh out several ideas for grant proposals, some of which are with people I met over there, which is always fun, and some of them are still pending, including one funded by Volkswagen. That's got to be hilarious. So I think it might be some diesel emission guilt. they're suddenly funding science. Anyways, if that gets funded, that'll be fun, because then I get to go back over there and screw around with them more. And I'll leave you with, oops, I'm sorry, before I do that. People I want to thank, people in my lab. So Gata and Cohen did a lot of the work that I was citing there in my little slides. Alan Suji helped out a lot with the initial parts of that project. These are fellow faculty friends that we basically steal stuff from because Malat has so many problems. So Felix was the guy I worked with. He was working in, Peter Becker is the chair of the department over there. And Nadine and Ashish were Felix's grad students. Nadine was the student who I handed this project over to. I think by the end there she was ready to strangle me, and so I don't know how much further she's moved on with this whole thing. But Ashish was relatively new, so maybe he can be bribed into working on this a little bit more. Funding sources, most important is all of you. Thank you for letting me go work overseas. It was a lot of fun, had a good time. NIH, LMU, KU, Cambridge, all these various people gave us cash. So thank you so much for all your financial support. And this is one of my favorite pictures of Munich. So if you've ever been to Munich, near in the southern part of the English Garden, there's a tiny little stream. When it flows into the park, there's a little weir that's set up there that creates this permanent standing wave. People surf on it all the time. You know, spring, summer, winter, you name it, they're always surfing there. And they have it all organized how they do that. People wait on the sides here, and then when this person falls into the water, someone else jumps in and goes. This goes on all the time. I was amazed at the number of times I would see people on the subway carrying surfboards. Right around the city, which is nowhere near any ocean, but they're trying to get down here to go surfing. So if you want to practice your surfing, that's the place to go. I'll end with one of my favorite little Munich phrases, which is servus, which translates roughly as aloha, meaning that it can mean hello or goodbye. So for me, it means goodbye. So thank you very much for listening.
That's good.
How do you spell my last name?
Your last name? Briggs, I spelled it wrong?
Yeah, no big deal. No, not in that one, in the credits earlier.
Oh, really?
Yeah.
Oh, that's bad news.
Oh, yeah, maybe on a, we just slowly Germanize you. Yeah.
That's bad news, because I think I copied that from
No, I checked PubMed. It's
okay, good. Good. But it's an easy problem to fix. Thank you. No, it's good. I have to go fix that. Yes?
Are you thinking of repeating all this stuff here? See if you could get it to work?
So, no, because I don't have a lot of the equipment that I need for that. It's simple to work with them because they have the mass spec that's right there as a user facility. And they don't have a lot of the equipment set up, and all the protein that I made for them is already there. I think it's definitely worth trying this experiment. But mass spec, this approach to mass spec, I believe has a very small success rate because there's so many things that go wrong with it and so much we don't know. So it's the kind of thing that it's fun to try a little bit, and if it works, great. If you're lucky, then you got something. But the amount of effort you put in to make it actually work, if it work at all, is probably not worth the cost.
What kind of mass spec was it?
I was never allowed to be anywhere near it, so I don't know. We were told to prepare our samples and we leave them in a certain place for the mass spec technician to deal with, because they were constantly afraid of different kinds of contamination causing problems. So I could look at it through a glass window, and that was it. So I know no more than that.
You said you had an initial success with the idea.
I had success in that I was able to run a gel, isolate out what I believe to be the complex I want, cut it out, soak it in crosslinker, and get something that would crosslink. I could do other things to show that it crosslinked. When we sent it off to the mass spec, though, when the data came back, there was no indication in the analysis that there were any crosslinks between ISWI nucleosomes there. That could be just because we had placed a little marker in the wrong place. We weren't aligned to see the complex. Or it could be that it wasn't working. But we did this a couple of times, it didn't work, and then I was flying home. But I sort of set up, here's the protocol you go through to actually do this, and try it with the rest of these proteins. Last time I talked with them, the problem was they'd gone through the different mutants we have, which is only 5, and none of them are working. So then the question becomes, do we troubleshoot why those 5 aren't working? Or are they not working just because of the geometry of the complex? Which we make another 5 mutants. Where to put the site in those. And this is what I kind of mean, where it gets to the point where I don't know how you can begin to rigorously go down this path without spending years and years and years on it unless you just get lucky. Yes?
Is this the Oktoberfest the whole Germany thing, or just the Baviarn?
It is a Bavarian, it's mostly a Bavarian thing. It celebrates this wedding that took place a long time ago. It was actually a big deal at the schooling my girls went to for the first few days they were there talking about that. We made a field trip. You think your field trips were great in school? Their field trip was to Oktoberfest, for God's sakes. That's where they got to go. A lot of places in Germany have decided, oh, we should Oktoberfest too, because we bring in drunk Australians and then we get money off of them. Many little tiny towns around Bavaria have some version of Oktoberfest, and a lot of the other big cities have it now, but the main one is still in.
The cross-linking is evidently a standard idea.
Yeah.
And it looks like a great idea to identify your sample in advance with the electrophoresis, with the gels. And the gels are a very standard technology. So you put together a new technology that could be used on a million different projects. And are you allowed to publish that as a method paper, as a concept paper? No. That's typical of biology. So why don't you explain that to us? In physics, if someone has a new idea, we're diagonalizing a 2x2 matrix. We're making a DC motor. They can publish that.
And what you're looking for, John, is novel.
Novel. It's not novel?
It's not novel.
But
If you,
how come
no,
you
let me
didn't know about it in advance?
I will explain to you how novel works. If you discover a mistake that someone else has made, let's say you repeat someone's experiment and get different results, and what they did wrong, you can't publish that because it's not novel, because someone else has already published the results. So that's the work you're in right there.
I want you to enlarge on that, because in physics, we think that failed experiments should be published. They're called limits.
Yes,
We have it.
we never published failed experiments in
We
biology.
think that ideas that have not been tested should be published. That's called theory. And in biology, the standards are you have to make a discovery for every paper, or
Yes.
whatever. How does it work?
You cannot publish a null result unless it's part of a larger context where you're, unless like maybe you have shown, like if someone said the equivalent of sky is orange all the time, you showed the sky is blue. You could say, well, I was able to show the sky isn't that orange, it's blue. As long as you have something else where you're adding on to the theory or changing it, that's fine. You can't write a paper that just says, these people are wrong, because I can show the experiment results here, they're wrong. That does not get published. Things have to be novel. What does that mean? It must be a new discovery you've made. So you have to literally move the field to an entirely new place, or caused, cured some disease, or whatever you want to do, in the most hyperbolic language you can possibly come up with. Otherwise, publish it. If you put too many equations in your paper, not publish it. And it's just, I mean, that's just.
True about nature papers, always.
No, but I mean, this is, but this is, Cohen can attest to this. This is the world it is, and there are many problems with this, but that's the way it is. What tends to be worse about this is not so much that it's hard to get good stuff published. The problem is it's easy to get crap published because people tend to overanalyze their data, or they tend to randomly conclude stuff from the data that's not there. And because there's been such a proliferation of journals of life sciences, it's not like there's hundreds. All you got to do is publish stuff, slightly changed in a few different journals, and then write a review about it all. And then you have a review paper where you say, well, this is how it's done. And then once that is set in stone, it's really hard to overcome it. This idea here, these two, well, this was a little bit of a fight. This was, years of fighting. And you'd be like, here is the data that supports our conclusion. Well, no, someone else said that's not right. But how do you argue our data is incorrect? Doesn't matter. So, yeah, so that's what it is. Well, we enjoy hearing about this. Thank you very much.